Okay. Am I audible? Good morning, students. Good morning, all of you. Okay. Yes, my voice is audible. Done. Okay. Okay. Come on. I will discuss the remaining part of genetics over there. Just I will talk about the content regarding to linkage and recombination. Linkage and recombination. We have already have some introduction regarding to this linkage and recombination. First of all, why did the concept of linkage has come? Imagine when they conducting Mendel's dihybrid cross, Mendel, according to the dihybrid cross, he has given independent assortment is the source of recombination. Independent assortment is the source of recombination. This was the word given by our Mendel. He said this. When we conduct dihybrid cross, where 9:3:3:1 phenotypic ratio we will get. In this, these two are recombinants, and this is a parental phenotype, this one is another parental phenotype. Then this was a surprise for Mendel, because when he was conducting monohybrid cross, there was no any phenotypic recombinants, but in dihybrid cross he could see phenotypic recombinants along with the parental phenotype. Monohybrid cross, law of dominance, law of segregation, that story was going well, but suddenly new concept has entered. How did recombinants are produced? Then Mendel said these recombinants are produced due to independent assortment of factors, which means in a, for example, these two are a pair of factors, these two are a pair of factors. Segregation of this pair of factors during at the time of gametogenesis, segregation of this pair of factors is independent to segregation of this pair. As a result of that, we are going to get four types of gametes over there. Capital Y, this will join with this capital R, small r, capital Y, small r, small y. These four gametes we are getting because this pair of alleles segregate independently of this pair of alleles. So that we are getting 16 genotypic combinations. There are four types. 16 genotypic combination. Phenotypically there are four types. One parental, another parental. These two are recombinants. This story was well established and everybody accepted. Mendel did extraordinary thing. Okay. Later, there were some people studying the same characters in sweet pea. That's called Lathyrus odoratus. When they were studying the dihybrid cross, same dihybrid cross which was conducted by the Mendel, not in pea plant but in another plant, that's called sweet pea, Lathyrus odoratus, and they got surprisingly phenotypic ratio 7:1:1:7. This is a parent, another parent. These two are recombinants. Mendel said these many recombinants are possible. How many recombinants you got? Only two. This is extremely low rate of recombination frequency. Then how could we understand this low rate of recombination? Several people studied and so many details were examined, and finally there was a person called as Morgan, Th Morgan. This person in Drosophila melanogaster conducted some experiment and mentioned this reduction of recombinants. I mean, Mendel said 6 recombinants we will get in dihybrid cross. But how many recombinants you are getting? Only 2 recombinants. What could be the possible reason behind the reduction of recombinants? Morgan mentioned the recombinants were reduced due to a concept that is called linkage. Now, the word linkage and the experimental verification that the linkage is happening in the organisms was explained by our Morgan. Then, how did linkage has entered in the field of genetics and what is the importance of the linkage? Imagine, Morgan mentioned independent assortment is true. He didn't say law of independent assortment that was proposed by Mendel was wrong. No. He said law of independent assortment is true when the genes present on, when the selected genes, selected traits or selected genes present on non-homologous chromosomes or two different chromosomes. This is the word was given by our Morgan. Then, what do you mean by two different chromosomes? For example, I have taken, this is a homologous pair. This is another homologous pair. Over here, I am studying about the genes. For example, capital A, small b, capital B. Here, P, Q, capital Q, small p. These are the genes I have selected. These two are homologous chromosomes. This is a homologous chromosome pair. Homologous chromosomal pair. Observe this. If I am studying about, observe the statement. I am conducting a dihybrid cross for the genes A and P. At the time, definitely law of independent assortment would be applicable. There is no doubt A and P will follow law of independent assortment. But imagine when I have chosen to study A and B, which are present on same chromosome, at the time, law of independent assortment most probably will not going to work out. In that case, why law of independent assortment is not possible if the genes present on same chromosome? We will call these genes are linked genes. Linked genes also produce recombinations by a phenomena called crossing over. Once again, imagine, look at this statement. I am writing a pair of dihybrid genes, capital A, capital P, small p. Observe this. This is a dihybrid. You should believe that. I have taken two genes present on two different chromosomes. Definitely they will undergo independent assortment. But let us take about this statement, capital A, capital B, small b. They are present on same chromosome. When there is a same chromosomal genes allocation happen at the time, the concept of linkage would enter in the field. Okay, because these genes present on same chromosome. Since they are present on the same chromosome, I am not saying they never undergo recombination. They will undergo recombination whenever there is a chance of crossing over. Now, if you talk about two genes which are linked genes develop recombination due to crossing over. Linked genes develop recombination due to crossing over. Now observe the story. If I take two genes present on the same chromosome, the two genes present on the same chromosome would produce recombinants, but by a process called crossing over. If two genes present on two different chromosomes, they do produce recombinants, but the phenomena is independent assortment. Okay, now I'm sticking to the concept of crossing over. What is crossing over? Exchange of chromatids, okay, between homologous chromosome, or exchange of the genes between non-sister chromatids of homologous chromosome. That's called as crossing over. Now here you wanted to make the crossing over. Now simple, crossing over frequency increases when distance between the two genes also increases. Observe, I'll take three genes on the chromosome A, for example, okay, C and E, small a, small c and E. These are the genes I have taken. Observe, I wanted to identify the crossing over between A and C. This much distance is available. And I want to see the crossing over between A and E. This much distance is available. More the distance available for the crossing over, higher the rate of crossing over. Obviously, distance would define the crossing over frequency. When crossing over frequency increases, the number of recombination frequencies will also increases. Now, what is the impact of crossing over increase and the linkage? Imagine when crossing over increasing, linkage obviously decrease. At the time, you need to define what do you mean by the linkage. Linkage stands for physical closeness of the genes. Closeness of the genes or the genes present on same chromosome. In order to understand linkage, physical closeness between the genes, let me take this example. ACE, all are linked or not? Yes, they are linked because they are present on same chromosome. But I will say linkage between A and C is more when compared to linkage between A and E. A and C linkage is more. Why linkage is more? Physical distance between them is less. What did you define the linkage? Linkage was defined like this. Physical closeness of the two genes present on the same chromosome. We will call it as linkage. When the two genes physically close or they are getting closer, then that linkage is going to be increase. More the linkage, lesser the distance between the two genes. That is a concept. If it is running like that, observe, you say when the crossing over is more, okay, and linkage should be less. Linkage should be less means distance between them is going to be less. At the time, observe the relation. When you talk about linkage increase, recombination What is the relation between linkage and the crossing over? Crossing over is responsible for recombination. Obviously, when the linkage increase, that means the distance between the two genes decrease, recombination is going to decrease. Okay. Now, I wanted to find out generally the distance between the, recombination chance based on the linkage. For that, what I do one thing, this distance between the genes, distance between the genes study by knowing recombination frequency. What do you mean by recombination frequency? That means how frequently the two genes can undergo recombination, or how frequently the two genes can undergo crossing over. Be careful. Wherever I use the term recombination, I am going to say they are going to have some sort of crossing over. Crossing over is the one which is making them to be recombined. Got the idea? Now, let me take recombination frequency. If I wanted to study, I'll have one formula, that is number of recombinants by total progeny. Number of recombinants by total progeny. That is recombination frequency. And I want to make it in the form of percentage. Then I will give it 100. Then one value I will get. After conducting recombination frequency, one of the student of Morgan has come to the new idea. If you know the recombination frequency, we can understand the relative distance between the two genes. That scientist name is called Stuart Evant. Then he said, if 1% of recombination frequency happened, that equal to 1 centimorgan. 1% of recombination is equal to 1 centimorgan. If you see 100% of recombination frequency, that is 1 morgan unit. This M does not stands for mole, that M stands for morgan unit. Then I will give one question based on this. Imagine, based on linkage and recombination, generally we see three model problems. One model problem, first of all, finding out recombination frequency with respect to dihybrid cross. Generally, I wanted to find out recombination frequency of a dihybrid test cross. Now find out recombination frequency and the distance between the genes. I have got 3 genes here. Okay, 3 genes. Observe the statement. The 3 genes, capital R, capital Y, or else they are giving 3 types of phenotypes. Okay, 3 types of phenotypes. Let me take capital R, capital R, capital Y, capital Y, capital R, small r, capital Y, small y, small y. Generally we go for dihybrid test cross. Next one, small r, capital Y, small y, small r, small y, small y. These are the genotypes, and relative phenotypes they have given like this. Find out. This one 120, this one is 100. Okay, and then this one you have got 60, and this one you have got what we call 100. Or else let me take easy numbers for you. Okay, here you got it is going to be, let me take 80. Let us take simple numbers. Here, remember, they have given dihybrid test cross values and they will ask you to find out the distance between the genes. I have already given one formula. There, simply go. Lesser numbers are called recombinants. More numbers are going to be parental combinations. Then observe 60 plus 80. Okay, 60 plus 80, 140 by 120. These two, 140 and also 100. Always you have to add a total number. And if you get the value you have driven, then you can get one value that's called a recombination frequency. That recombination frequency is nothing but the distance between the two genes. Okay, this is one kind of model problem. They will give these values and they will ask you to find out the recombination frequency between the two genes, or simply what would be the value of the distance between the two genes. This is one model problem. Next one, another model problem. They will say two genes, another model problem I am giving, two genes A and B are linked. A and B are linked. Okay, they showed 20% of recombination, 20% of recombination, 20%, they showed 20% of recombination. Then what is the each parental frequency? What is each parental frequency? Come on. They are saying total 100% if you consider. They say recombinants are 20%. Be careful. In dihybrid test cross, we are going to get 4 types of phenotypes. Monohybrid test cross 2 types. Dihybrid test cross, we are going to get 4 types of phenotype. Out of the 4 types, 2 are parental phenotype, 2 are recombinants. They already say 20%. How many recombinants? 20%. How many types of recombinants? Percentage of the recombinant is 20%. How many types? You will get 2 types. So each recombinant 10%. Okay, and parental combination, you have already mentioned 20% recombination means 80% parental combination, and each parental one equal to be 40% in total. For example, we will get 4 types, 10 recombinant, another recombinant, 40, that is parental, another parental one. Do you remember in dihybrid test cross we have got 1:1:1:1:1. This is the result what we need to get, but that result will not be in the form of 1:1:1 ratio. You will get different values if they are linked genes. This is another kind of model problem. Simple. They will give you some genotypes and they will ask you to identify, okay, what type of members are parents, or what is the parental frequency and what is the recombinant frequency. I have given here theoretically. I will give examples related to the genotype. They say A and B are linked. A, small a, this is their genotype. Okay, and if A and B are linked means they are present on same chromosome. A, B, small a, small b. Now parental combinations A, B, small a, small b. A, B is one variety exactly. Okay, and let us consider A, B, B, one variety. Okay, and recombinants you will get capital A, capital B, small b, one variety, small a, capital B, small b, small a, small b, small b. This is one parent, another parent. These two are recombinants. Remember, most probably they will ask you to find out recombinant gametes. At the time, this is I'm talking about test cross result. Next, I will go to recombinant gametes. Tell me how many types of gametes it will produce. I have a genotype. Come on. I have a genotype A, B, B. How many types of gametes it will produce? It will produce 4 types of gametes. What are they? AB, A, small b, okay, small a, capital B, and small a, small b. This is a parent, this is a parent. These two are recombinants. What is the frequency of this one? According to my question, 10%. What is the frequency of this one? 10%. This one is 40%. This one is another 40%. That's it. Okay. This is about second model. First model, they will give distance between the genes. First of all, we need to find out. If they wanted to ask you to find out the distance between the genes, they will give few test cross results. In that, randomly choose who are recombinants. Recombinant values are less, parental values are more. Okay. Next one, this another one, they will give like this: 3 genes. Another model, 3 genes A, B, C are linked, and A to C distance is 10 centimorgans, B to C distance is 20 centimorgans, and next one, AB distance is 10 centimorgans, and find out the sequence of them. This is another model. If they are present on the same chromosome, it's easy to find out. First, you go one thing, find out the longest one. Out of that, find out the longest one. Who is the longest one distance? B and C. Okay. Now observe next one. I know B and C. Let us take A to B. A to B is what? 10 centimorgans. I can keep A this side. At the time, B to A become 10 centimorgans. If they did not give the value A to C, I can keep A either this side or this side. Either one I can keep. But they have already given A to C distance is 10 centimorgans, so that I can keep B, A, C. So the sequence of the genes which are present on the chromosome should be B, A, C. That's it. These are the only 3 model problems that we will get very frequently on the basis of linkage and recombination. This is the concept of linkage and recombination. Okay. Next, I will choose few more fundamentals regarding to molecular basis of inheritance. Molecular basis of inheritance. Few more, just the things which are related to molecular basis, where we confuse some of the things, and also what do we need to understand. Remember, this is a polynucleotide chain. In this polynucleotide chain, how many nucleotides are there? 4 nucleotides. Am I correct? 4 nucleotides are present. If they are having 4 nucleotides, now how many phosphodiester bonds? This is one phosphodiester bond, another phosphodiester bond, 3. Imagine, sometimes what they will ask, how many phosphodiester bonds, how many glycosidic bond, how many ester bonds, etc. they will ask. Let us have some small kind of calculation problems there. Here, if I take n number of nucleotides present in a polynucleotide chain, for example, n number of nucleotides, tell me how many phosphodiester bonds are present? Phosphodiester bond. How many? Simple. If n number of nucleotides are there, simple. 4 are there, 3. So n minus 1 number of phosphodiester bonds are there. Next question they are asking, how many glycosidic bonds? Glycosidic bonds. How many glycosidic bonds? Every nucleotide has one glycosidic bond. Am I correct? So, 1, 1, 1, 1. This bond is glycosidic bond. How many nucleotides are there? Those many glycosidic bond, obviously. Okay. Next, they are asking phosphoester bonds. Phosphoester bonds. How many phosphodiester bonds you have calculated? How many phosphodiester bonds you have calculated? Each phosphodiester bond is having two phosphoester bonds, but one more ester bond is extra, which means already you have calculated diester bonds 2n minus 1. Each diester bond is having two ester bonds plus 1. Over there 2n minus 2 you will get. So 2n minus 1. These many numbers. Is it okay? This is the formula to calculate phosphoester bonds. This is for one strand. Copy if you need, otherwise okay, let it go. Here next one. I have seen many students are confusing about Chargaff rule. What is Chargaff rule? Chargaff rule says there was a scientist called Erwin Chargaff. When he studied so many nucleic acids, he come with new idea. That is, when you talk about Chargaff's rule, he mentioned in double-stranded nucleic acids, Na stands for nucleic acid. In double-stranded nucleic acids, purines ratio is equal to pyrimidines. How many purines are there? Those many pyrimidines. What do you mean by this? If you take nucleic acid, either it's a DNA or RNA, only Chargaff rule applicable to double-stranded nucleic acid. I mean, you have single-stranded nucleic acids also. There is no complementarity between the nitrogen bases in case of single-stranded nucleic acids. Only double-stranded nucleic acid will have the concept of complementarity working. Okay. Now over there, you take double-stranded DNA, double-strand RNA, doesn't matter. Chargaff rule will be applicable. How many types of purines present in DNA? A and G. How many types of pyrimidines present? A pairs with T, so G pairs with C, these two. This is the same thing I will write like this. A plus G by T plus C is equal to 1. These two are not two different statements. They are same statement I have written in two variations. Okay. Then come back to another one. This is what Chargaff has mentioned. Then one more thing. When you talk about the same detail, when you talk about double-stranded nucleic acid, A is equal to T. I am writing it is equal. How many A's in one strand? Equal to number of T's in another strand. G is equals to C. How many G present in one strand? Obviously equal to C of another strand. Altogether, A plus T by G plus C, it is not equals to 1. Be careful. A and T base pair. Base pair it is. G and C is another base pair. Purines by pyrimidine is equal to 1 according to Chargaff. But Chargaff didn't mention one base pair is equal to another base pair. This is a base pair. Its ratio never equals to another base pair because base pairs will have different numbers in different types of species. Got the idea? So A plus T by G plus C is not equals to 1. This is about the phenomena called Chargaff rule that would be applicable to double-stranded nucleic acid. Next one. When we talk about a double stranded nucleic acid, the diameter of the double stranded nucleic acid, that means the distance between one strand to another strand, always constant. That would be around 20 angstroms or 2 nanometers. The diameter is always constant. Now, when you talk about the distance between the two strands is always constant, why the distance is always constant? The reason is this. If you take a purine, purine has two heterocyclic rings and the pyrimidine has one heterocyclic ring. In one base pair, how many heterocyclic rings? Obviously, 3. When 3 are there, that is constant. You take G and C, 3 heterocyclic ring. You take A and T, 3 heterocyclic ring. That is making always entire, okay? Always entire length of the DNA strand would have one purine come opposite to pyrimidine, one pyrimidine will come opposite to purine. Purine never pair, pyrimidine never pair. As a result of that, always in the center of the double stranded DNA, purine come opposite to pyrimidine. That is making the diameter of the DNA is always constant. Those details you can understand. This is called central dogma. What do you mean by dogma? Dogma. Dogma means a rule. There is no any relation with dogma and dog. Okay? Dogma means a rule that does not going to be changed frequently. There was a person called Crick. Crick proposed central dogma of molecular biology, which says wherever there is a nucleic acid, in the nucleic acid to other biomolecule, flow of genetic information is like this. Genetic information flow from DNA to DNA. We will call it as replication. Genetic information can flow from DNA to RNA. We will call it as transcription. Genetic information can flow from RNA to protein. That's called translation. Rarely, genetic information even flow from mRNA to DNA. This is called as reverse transcription. Reverse transcription. This was identified by scientist called Timin and Baltimore. Timin and Baltimore. That's why it's also called Timinism. This is a reverse transcription. That's rare. Next one. Packaging of DNA. Okay. These things. Yes, injected into the mice. Mice died. And we have heat killed. Yes, mice alive. R injected into the mice. Mice alive. Heat killed. Yes, and R injected into the mice. Mice died. And final conclusion. There is an entry of DNA from or genetic material from heat killed S strain to live R strain. This detail was given by Frederick Griffith, that you can fathom or you can understand it. And finally, only some details. The red indicate radioactivity. Okay. Now final conclusion in Hershey Chase experiment. When we talk about those bacteriophages which are radioactive with DNA, that DNA has entered into the bacterial cytoplasm. If the material that could not be entered into the cytoplasm, which could never be act as genetic material. Okay, so protein is left over on the top of the bacteria. Genetic material is entered inside the bacterium, so that the one which is entered inside the bacterium is the one which is responsible for acting as genetic material. That is a conclusion by Hershey Chase experiment. These are the properties. Okay, and we can understand finally. Remember here they are saying there is a concept is there, RNA world. Imagine there was a scientist called Alexander Rich. He was an evolutionary biologist. He proposed one concept that is called RNA world. What is this RNA world? RNA world says like this. During the course of evolution, the first genetic material was RNA. Later, from the RNA, DNA came. From the RNA, proteins came. How could you prove that first genetic material during the course of evolution is RNA? One more thing. RNA world is not exactly the concept of molecular biology. RNA world is a concept that is having some connection with evolutionary biology. In the beginning of the cells, for example, whether it's a eukaryotic or prokaryotic cell, in the beginning of cells, the first genetic material was RNA. How could you prove the first genetic material was RNA? They are showing some evidences. If you are talking about RNA was the first genetic material, there are evidences. Some metabolic reactions controlled by metabolism is controlled by RNA. What are those metabolisms controlled by RNA? Imagine, we know metabolism is nothing but anabolism, catabolism. They are controlled by variety of enzymes. Enzymes sometimes required coenzymes also. Metabolism is under the control of enzymes and coenzymes. Observe this idea. If you are talking, how did RNA can able to control metabolism? RNA controls metabolism in this way. First one, RNA act as coenzymes. Some of the coenzymes I will notice here who are having RNA. Best one, acetyl CoA. Another one is NAD, nicotinamide adenine dinucleotide phosphate. That nucleotide present in NAD is coming from ribonucleotide. And FAD, flavin adenine dinucleotide. They are also responsible for RNA derivatives. I mean, NAD is a RNA derivative. RNA is the one which is giving NAD. FAD is also derivative of RNA. Think about that. If NAD and FAD and acetyl CoA removed, there is no concept of Krebs cycle. There is no concept of electron transport chain. Nothing would be operated. That's the reason why, in order to operate metabolism, directly RNA molecule is required. Next one, how did RNA involved in translation? During at the time of translation, there is a ribozyme called 28S rRNA and also 23S rRNA. These two components act as ribozymes. They are the one responsible for the formation of peptide bond between the two adjacent amino acids, so that we will have translation process. Think about that. If there are no ribozyme means RNA act as a catalyst. If there are no ribozyme, there is no chance for the formation of peptide bond. So RNA is highly essential as a catalyst during translation. Then what about the thing that is splicing? If you talk about during the splicing process, there is a component called snRNA. snRNA. Who is snRNA? Small nuclear RNA. This is the one which is actually making splicing, that is cutting off intron, joining of exons. That phenomena is under the control of our snRNA. snRNA is also doing splicing control. Translation is also controlled by RNA. Metabolism is also controlled by RNA. In this case, RNA can act as both catalyst as well as genetic material. During the course of evolution, RNA has some sort of chemical difficulties in order to maintain its structure. It was chemically unstable, and as a result of that, it has gone some modifications. From that RNA, DNA entered, and from the RNA there is another molecule entered, that is called protein. Like this, they are concluding the first genetic material during the course of evolution is RNA. Next one, replication. You know the replication process, origin of replication, where the replication is going to be begin. The mode of semi-conservative mechanism, where both strands act as template. One strand will act as template for one new DNA. Another strand would act as template for another new DNA. Those concepts we can able to understand. Okay. And here also, I have seen many students are getting confused over here. This phenomena, Meselson-Stahl experiment. First, we are growing DNA in N15 medium. Be careful. N15 is not a radioactive medium. Okay, it is a kind of isotope which gives some molecular weight differences. We will grow E. coli in several days in a medium that contain N15. Not only the DNA, all the nitrogen containing components would be having some sort of radioactivity. Once the radioactivity inserted in the E. coli, then we will transfer it into the normal medium, that is called N14. Keep it in mind, N14 is not a isotope. N14 is just a normal. N14 medium is not an isotopic medium. Then you have taken the DNA which is labeled with N15 and given to N14 medium. N14 medium, there is no any supply of N15 hereafter. That means whatever the DNA that is newly synthesizing, that DNA must be having only N14 availability. N15 availability is not there. That's why newly synthesized DNA is going to have, after, why they have mentioned 20 minutes? In order to make one bacteria to become two, we need 20 minutes. For every 20 minutes, one bacterium is going to become 2 copies. So this DNA is called hybrid DNA. What is hybrid? One strand is N15, another strand is N14. After 20 minutes, 100% of the DNA they noticed hybrid. And after 40 minutes, 50% of the DNA is hybrid. Or in simple ways to say, after 40 minutes, out of 4 DNAs, 2 DNAs were hybrid and 2 DNAs are lighter. After 60 minutes, 2 DNAs are hybrid, 6 DNAs were lighter. Like that it continued in every generation. Hybrid DNA was constant. Always number of hybrid DNAs are 2. Why we are not getting hybrid DNA any more expansion? Because whatever the 2 DNAs that you have taken from heavy medium, the same DNA strands are going to be continued. Hereafter, there is no synthesis of heavy DNA. So, how many times you are replicating? Those many times, for example, you have replicated in n number of times. 2 to the power n minus 2 lighter DNAs are going to be present. Lighter DNAs are present. This is Meselson. This is the only thing. Don't make complicated. Just they have asked 80 minutes or 100 minutes or whatever it is. First, every 20 minutes DNA number is going to be double. For example, they have given after 100 minutes how many hybrid DNAs, lighter DNAs are present. 2 to the power 5 minus 2 lighter DNAs. If you take 100 minutes, 100 by 20, okay, or 1 hour you have taken, that is 60 minutes. 60 minutes, how many times bacteria can be multiplied? 3 times. 2 to the power 3 minus 2, that's it. It's an easy thing. Don't make it complicated. Then after that, replication machinery. Be careful. This replication is replication fork. This is where the area where there is a presence of the enzyme helicase, which is breaking hydrogen bond. One strand would be synthesized continuously towards the replication fork. That's called leading strand. And another strand will be synthesizing discontinuously away from the replication fork. This is called lagging strand. Lagging strand. Always this rule never change. Leading strand continuously synthesized towards the replication fork. Lagging strand is going to synthesize away from the replication fork discontinuously. Clear the statement. So that lagging strand which is synthesizing discontinuously away from the replication fork will have small fragments. Those fragments are called Okazaki fragments. Okay, Okazaki fragments. Okazaki fragments later will be joined by DNA ligase. Now tell me, I have taken leading strand. In which direction it will synthesize? It's synthesized in 5' to 3'. Lagging strand will be synthesized in which direction? Lagging strand is also synthesized in 5' to 3'. Why it is like that? Because always a polynucleotide chain will be synthesized in only one direction. That should be 5' to 3'. Another direction is not possible. If another direction is not possible, then this strand also might be synthesized towards the replication fork. If they are asking why DNA replication is discontinuous or semi-discontinuous, be careful, DNA replication is semi-discontinuous because one strand is continuous, another strand is discontinuous. Why DNA replication is semi-discontinuous? Because always is polynucleotide chain will be polymerized in the direction of 5' to 3'. A polynucleotide chain never polymerized in the direction of 3' to 5'. That's the problem we have. One strand continuous, another strand discontinuous. One more thing, where I have felt some students are confusing repetitively. Imagine if I talk about DNA replication. DNA replication does not start randomly. It starts at a specific point that's called origin of replication. Whenever the DNA helicase joins to origin of replication, this is the point. Okay? DNA is going to open at the location of origin of replication. This is called one replication fork. This one is another replication fork. When you talk about replication forks, from the origin of replication, we have two replication forks. Is it okay? Now over there, one replication fork will continue this direction. Another replication fork will continue the DNA replication this direction. Like this, from the origin of replication, or you see, DNA replication is bidirectional. DNA replication is bidirectional. It's not always it is possible, but generally in case of eukaryotes and prokaryotes, replication is bidirectional because from the origin of replication, at least two replication forks will be formed. Each replication fork will run opposite to another. That is making DNA replication is bidirectional. Then transcription, conversion of DNA to RNA, we call it as transcription. Observe, in a typical transcription unit, we have the following parts: promoter, terminator. Imagine, this is the exact part which is going to be converted in the form of mRNA. For example, that mRNA is going to be converted in the form of protein. The part of the transcription unit which would be converted in the form of protein, generally we call it as cistron. What do you call that? Cistron. Be careful. Cistron is a part of the DNA that is converted into polypeptide. If I say cistrons present in transcription unit, correct. Do all transcription units will have cistron? No, because there are some transcription units, they have promoter, terminator, structural gene, but that structural gene does not convert it to mRNA. It may be converted to tRNA or RNA, or variety of other RNAs will also going to come. See, all structural genes are not having cistrons, but definitely cistrons are present in structural gene only. A structural gene which would be converted into mRNA and eventually which converted into protein, we will call it as cistron. Then once they identified the word cistron, the DNA which can code for polypeptide, there was a noticing of a statement, that is, when you talk about cistron, in case of generally eukaryotes, you said structural gene is nothing but a part of DNA that is can be converted in the form of protein. Well done. But does the entire DNA is going to be converted in the form of a protein? Then they notice in a cistron, especially in case of eukaryotes, There may be a presence of exon, there may be a presence of intron. Exon is called as functional part, that is which would be converted in the form of protein. Intron is nothing but non-functional part. Again, observe. In a transcription unit, all transcription units will have structural genes. But some transcription unit structural genes contain cistron. That cistron is nothing but a structural gene which is coding for polypeptide. Now, if I take cistron, entire cistron is not functional in case of a eukaryotic gene. Such kind of genes you have to call them as split genes. Split genes. Observe, the split genes are those genes that are having some part is functional, some part is non-functional. Got the idea? Altogether, when you observe cistron, cistron is having some part is functional, some part is non-functional. In case of prokaryotes, entire cistron is functional. In addition to that, in one transcription unit, you may have more than one cistron. That's why prokaryotic genes are called prokaryotic transcriptional unit will have an mRNA. That mRNA can able to produce many cistrons, or the mRNA can produce many polypeptides so that it has many cistrons. That's why in case of prokaryotes, genes are non-split, and they are also called polycistronic genes. Polycistronic. In eukaryotes, the genes are split, but they are monocistronic. Okay, this is about the concept one would need to learn regarding to structural gene. Remaining things you know, promoter, terminator, etc., etc. Next one, transcription unit. The same words what I have explained they say here. Observe here they are saying inheritance of a character is also affected by promoter and regulatory sequence of a gene. Remember, when we were talking about genetics, you might have learned one statement that is called, what is the definition of gene? Tell me what is the definition of a gene? If you could not able to directly say to me, or you say to yourself, what is the meaning of a gene? Gene is nothing but unit of inheritance. Am I correct? Okay, gene is unit of inheritance. First of all, what is inheritance? Let me say one thing. We will understand inheritance like this, simple one. I will take a red flower from a female parent, crossed with a white flower from a male parent. I have got the result is red flower. All 100% flowers in the generation were red flower. Okay? Think about this. When we consider red and white over there, I have taken two parents. One parent is red, another parent is white. Then here I could see only red. That means female parent phenotype inherited, male parent phenotype is not inherited. What do you mean by that? Inheritance. If a character that could be seen parental generation as well as in the progeny, then only I will say the character has been inherited. Something that is inherited means it should be present in the parent, and also that character I should see in progeny. Then only the character is inherited. What happened? I don't know. But if I could see the character in parental and progeny, both ways I could say the character is inherited. Is it okay? Imagine a statement. There are two parents. Father dark skin, mother light skin, and they have a child who who is light skin. Mother character inherited, father character did not inherit. Are you getting the idea? I'm not saying father genes are not there. I'm talking about character. Okay? Genes definitely may come, but whether the gene has come or not, how do I justify? Only by observing the phenotype. Yes or no? Think about that. There is a transcription unit which has promoter, terminator, structural gene. Now, we know already there is one concept, that is, how do we convert dominant genes to recessive? What was the process that dominant genes converted into recessive? By a process called as mutation. If some kind of mutation happen in the structural gene, obviously the phenotype will become defective. Okay, I know that if something happened to structural gene, since it is converted in the form of protein, and the protein will also going to be defective altogether, structural gene defect will leads to change in the phenotype. Okay, done. Structural gene is responsible for inheritance. Think about that. There is a problem happened in promoter. If promoter mutated, then also the gene inheritance would be affected. If terminator mutated, the inheritance of the gene could also be affected altogether. Okay, whenever there is a transmission of a character from parents to progeny, if that character is not inherited, always we cannot blame structural gene. Sometimes the defect may be in promoter, sometimes the defect may be in terminator, sometimes there may be defect in operator. So many areas would be defective. So whenever we talk about inheritance of a character, transmission of a character affected by promoter and other regulatory sequences also. Okay, hence sometimes regulatory sequences loosely defined as regulatory gene. Loosely defined means typically. Generally I can say promoter sequence is a gene, or else promoter gene, terminator gene, operator gene. I can say. Why I am saying they are gene, they are not converted in the form of polypeptide? It's not compulsory for every gene to be converted in the form of polypeptide. When do you call a sequence of a DNA is gene? If it control inheritance. Does the promoter can control expression of a gene? Of course. So it is a gene. Does the promoter control expression of the particular area in other along with other? Along with the protein, along with the promoter, operator, terminator, and some other sequences also must be noticed as genes because they control inheritance. Keep it in mind. Then types of RNAs, etc., etc. Here they are saying one thing is here exactly. In our NCERT book, they have given the sequence of post-transcriptional modifications of hnRNA. What are the modifications the hnRNA can undergo? It will undergo the modifications like capping, tailing, splicing. Am I correct? Tell me the sequence of them. The sequence should be first capping, after that tailing, after that splicing. Splicing will occur in last. Is it okay? Capping, 5' end protection. Tailing, 3' end protection. If cap and tail and removed exon, that part is called mRNA. That mRNA will be transported from nucleoplasm to cytoplasm and subsequent changes will occur for translation. And here they are saying split gene arrangement, probably an ancient feature of a gene. Split gene arrangement, probably an ancient feature of a gene. What do you mean by split gene? A gene that it has introns and exons together is called split gene. A gene which has introns and exon together we call it as split gene. Am I correct? In that case, imagine a statement. I have a gene with introns. I can notice probably this gene is an old gene. I am not saying a gene without intron is new gene. But I have this much confirmation. If a gene that is having intron, probably that gene is a old gene. How can you confirm a gene with introns is probably a old gene? Simple. Whatever the genes during the beginning of the evolution given to an organism, these genes gradually modified. Is it okay? They are modifying gradually. That modification has happened in the name of mutation. That mutated part still kept in the gene, but that gene still functional. Careful, one thing I will say. Imagine in the beginning of evolution, when we were coming from our ancestors, Neanderthals, etc., when we were coming at the time, we used to had thick body hair, body fur we used to had. But when we started to use clothes and protecting our body from the external things, and that fur gradually started to decrease. Think about that. When we came from our ancestors, at the time, there was a gene that is responsible for controlling of our body hair. That gene still is there in our body, but it is not expressing as it was expressing in beginning of the evolution. Do you say the hair producing gene on human body is not expressing now? Still it is expressing, but it was not expressing like this. It was expressing in earlier days of evolution differently. Are you getting the idea? Now, those genes which are gradually modifying during the course of evolution have got certain regions which are in the form of inactive portions. Some region they are still maintaining active portion. That active portion is called exon. The inactive portion is called intron. Altogether, split genes, I can say, they were old genes because they have some old parts. What are those old parts? The old parts are nothing but introns. So, split gene arrangement probably represent an ancient feature of genome. Split gene probably represent an ancient feature of genome because introns are reminiscent of antiquity. Introns, which means those portions of the DNA who are not expressing right now might be the reminiscent. Reminiscent means remainings of antiquity. Got the idea? This is about what they want to say there. And after that, genetic code. You remember these codes? Okay? You have to remember. In the class here and there I might have given some clues to remember all those things. You follow up that. Okay? You have to remember. Degeneracy. Tell me, what are the amino acids will have highest degeneracy? There are 3 amino acids in the genetic. What do you mean by, first of all, degeneracy? What is degeneracy? Sometimes an amino acid could be coded by more than one codon. Degenerous. Generous means faithful. Degenerous. Now one thing, tell me. One amino acid coded by more than one codon. Okay? Next one. One codon always codes for one amino acid. Out of these two, which statement is correct? Amino acid coded by more than one codon. One codon always code for one amino acid. You should remember both statements are correct. Imagine here there is a codon and amino acid. For every codon there is an amino acid. Okay? Codon, it's representing amino acid, we call it as genetic code. Well done. Come back to the statement. A codon coding for an amino acid. Okay? Codon always code for one amino acid. But this amino acid is always not coded by this codon. This amino acid will have some other kind of partner. In that scenario, out of the codon and amino acid, who is degenerous? Who is not faithful? Amino acid is not faithful. Codon is always faithful. One codon always code for one amino acid. There is no codon which can code for more than one amino acid. But there are many amino acids which are going for more than one codon. So I can say there is a degeneracy present in genetic code. This is overall is called genetic code. There is a presence of degeneracy in genetic code. Am I clear the word? Then in this case you said some degeneracy is present for some amino acids over there. In this case observe serine, arginine and leucine. They are highest degenerative. You can't identify which codon coded by the serine because serine has 6 codons. Arginine is also having 6 codon. Leucine is also having 6 codon. Highest degeneracy. Highest degeneracy. And then go to, see, wherever there are huge number of people degenerates, 1 or 2 people will be generous. Okay? The generous amino acids are tryptophan. Tryptophan, only one codon, UGG. And also our well-known one, methionine, AUG. These two amino acids are no degeneracy. For these 2 amino acids, there is no concept of degeneracy at all. Highest degeneracy 6. No degeneracy, that is 1 codon, 1 amino acid. That is tryptophan and methionine. And there will be odd number 1% is the field. Isoleucine. Out of all amino acid, isoleucine is the only amino acid that is coded by 6 codons. Sorry, that is coded by 3 codons. 3 odd number. Remaining all, some will have 2, some will have 4, some will have 6. Okay? 4, 6, 5 will never be there. There is no amino acid that is coded by 5 codons. Got me? And universality, etc., etc. Mutations. This is well-known concept. You study. Here, tRNA. tRNA was act as adapter molecule. When you talk about adapter concept, this concept was introduced by Crick first time. What did Crick say? He said, during translation, there should be someone who can bring amino acids by knowing meaning of the codon. Think about that. I want to translate English to Telugu. There is a theory that they have given in English and I want to translate it into Telugu. At the time, I should know English as well as Telugu. If I know only Telugu, how can I translate? Not possible. So, obviously, there is some molecule in the cell which can, one hand it could read codon, on another hand which will bring amino acids for the translation. This component is called adapter. That adapter concept first time given by Crick. Before this adapter concept recognition, tRNA was considered as some kind of RNA which is very small, running here and there. Okay? And they used to call the tRNA as sRNA. S stands for soluble RNA or small RNA which is running without any significance in the cytoplasm. They thought this about tRNA. Later they recognized this is not small part. This RNA is very important, that will be helping for bringing of amino acids to translation process. So first they recognize adapter, and the adapter was not initially thought our tRNA. Later tRNA has adapter nature they recognize, and from that moment onwards tRNA is called adapter molecule. The three-dimensional structure of tRNA typically mentioned as inverted L shape, and the two-dimensional structure will be mentioned as clover leaf model. And translation process. Here I have seen many students are, imagine, this is a kind of translation procedure in the middle. Okay, incoming tRNAs will bind to A site and bind to P site. For example, one tRNA has come to A site. If it has complementarity between its anticodon place and the anticodon present on A site, that tRNA will bind to A site. As a result, the tRNA will fix on the A site, and there is a formation of peptide bond between A site amino acid to P site amino acid. Exactly to say, P site amino acid to A site amino acid peptide bond is going to be formed. Once the peptide bond is formed, okay, then the ribosome started to move. How they will move? They will move from 5' to 3' exactly one codon distance. One codon distance. In this direction they will move one codon distance. If they move one codon distance, A site will become empty for one more tRNA. A site already existing tRNA will come to P site. The P site one will come to E site and then exit. Like that, whenever there is a translocation, incoming amino acids with tRNAs will bind to A site, and there is a formation of peptide bond between P site and A site, and the tRNAs which are empty or uncharged, they will be eliminated from E site. And regulation, this is easy, you can understand. Imagine there is a concept is there, that is regulation of lac operon. Okay, when we talk about regulation of lac operon, in our NCERT book, how lac operon will be regulated negatively? Be careful. Negative regulation, positive regulation will also there. What do you mean by positive regulation? Positive regulation means increasing of expression. Increasing of expression. Expression must be more than normal. Negative regulation, less expression. That is called as negative regulation. Now in this case, how does lac operon will be negatively regulated? Imagine lac operon has certain genes, ZYA genes, we know that. Z gene stands for beta-galactosidase, Y gene stands for permease, and A gene stands for transacetylase. What they do? They all together involved in a phenomena called as breakdown of lactose. If so, whenever lactose enter, the genes need to be expressed. When the lactose is not there, the gene should be suppressed. In that case, imagine, in the absence of lactose, okay, then lac operon genes are not going to be expressed. That phenomena not to be expressed is called repression. What do you call that? Repression. When repression is happening, then repressor will go and bind to operator position. Repressor is coming from I gene, that's called regulatory gene. And then RNA polymerase could not able to access ZYA genes. Next, when they added lactose act as an inducer. The lactose will go and bind to repressor, and the repressor will undergo some conformational change, and it cannot able to bind to operator site. And the operator free, RNA polymerase can able to transcribe them. Okay, altogether. Only when there is a presence of lactose, the genes are started to express. When the lactose is not there, they will undergo repress. This phenomena, in the absence of lactose, lac operon genes undergo repression. This phenomena is called negative regulation. Okay, rarely they may even undergo positive regulation. You need not to touch it. That's about lac operon. Up to this enough. We will meet in another class. Okay, that's fine. Well done. Okay.